Good afternoon, everyone. Thank you for joining us this afternoon for our Cholangiocarcinoma Foundation webinar. We're so happy you're here. Today, we're pleased to be joined by the team from Sengen. I just wanted to remind everyone that you can put questions in the chat box throughout the presentation, and we will do a Q&A session at the end and get your questions answered. So feel free to type your questions in as they come, and we will get to them as soon as possible. And for now, I will turn it over to Carla to introduce her team.
Great. Thank you, Melinda, and the Cholangiocarcinoma Foundation for inviting us to tell you about our new diagnostics. Again, I'm Carla Grandori, and I am the founder and CEO of Sengen Precision Medicine. The company spun off from the Fred Hutchinson Cancer Centers, where I was a researcher for over 25 years. Again, I'm not a physician, but a practicing molecular cancer researcher. And we and other co-founders, they were oncologists from the Fred Hutchinson and computational scientists, decided in 2015 to put at the service of patients our best knowledge, technology, to really enable to personalize cancer treatments. And today, our speaker will be Dr. Payal Chatterjee, who also was trained at the Fred Hutchinson Cancer Center. She is an expert on cancer pathways and molecular targets for cancer. And she has taken what was my role to prepare the report of the Paris test for every patient. She works under also with the help and supervision of our CLIA laboratory director, as our test is CLIA certified across the United States. With that, I also is joining us today, Maddie Polastro. She also comes from the Fred Hutchinson Cancer Centers, trained as a clinical coordinator. She actually has also taken on the responsibility to manage our collaboration and clinical studies. She has created a really smooth workflow to enable the receipt of samples and transfer from various hospitals across the country and sometimes from overseas all the way to our laboratory in Seattle. With that, Dr. Chatterjee today will tell you about our experience working in cholangiocarcinoma, as well as across many other cancer types. And feel free to ask questions. I hope that you will find it informative.
Hello, everyone. Can everyone hear me well? I think so.
Yes.
So as Carla already introduced our team, I am Payal Chatterjee. I'm the lead scientist of Paris test here at Sengen. And I'm going to give today's webinar titled Personalizing Cancer Treatments with the Paris test for cholangiocarcinoma patient. So I'm going to give a brief overview of how Sengen was founded and what are our workflow for a sample collection to the report generation, as well as I'm going to give I'm going to tell you that what are the strengths of the Paris test and why we think it's a game changer in the field of cancer therapeutics. I'm also going to give a small snippet of our cholangiocarcinoma patient cohort and present an exceptional case to you. Oh. Somehow I'm not able to progress 2 slides.
You might need to click over on your presentation and try.
Okay, thanks. So every individual cancer is unique, and even in a particular type of cancer, such as cholangiocarcinoma, where we see certain gene mutations are common, but the whole genomic mosaic or the genomic constellation is different for each patient. It's never same from one patient to another. That's why personalized precision medicine treatment is needed for every patient. So that's the idea basically behind the Sengene's Foundation. Sengene founders and innovators, as Carla mentioned, were in Fred Hutch Cancer Research Center. They, through a rigorous research method, they established a protocol for organoid derivation from patient's tumor. And through the help of the robotics, they established a technique for drug screening on those organoids. And with the help of the revolutionary analytics, treatment were ranked. So all the drugs, what is tested in Paris test, can given a rank from exceptional to no response, giving the doctor or the oncologist.
We're watching videos.
Is everything okay?
We're good.
So giving the doctors or the oncologists the power to make an informed decision for future treatment choice. So CLIA, so our Paris test is a CLIA certified first-in-class phenotypic pan-cancer assay, which can be used for all solid tumors. And it gives the oncologist power to basically choose the best treatment possible for a patient based on our patient-specific drug sensitivity results. So why organoids? So organoids are 3D structures which mimics the human cancer best because it actually maintains the tumor heterogeneity and physiology of any human cancer. So in Sengene, we have cancer-specific drug panels, which is almost up to 43 drugs. And these drug panels were designed as per the standard of cancer treatment, standard of care treatment for that particular cancer. According to the genomic landscape of that cancer, the FDA-approved chemotherapeutics or targeted therapeutics for that particular type of cancer, as well as any available clinical trials. So Paris test can identify the candidate therapies in patient where either no genomics is available or where no known druggable genes or the biomarkers are available. And we have seen a strong concordance with the genomics as well as with the retrospective clinical evidences, such as like whatever treatment patient has received before and progressed, we see the resistance. And also we see a very strong prospective clinical evidences in our cohort, whatever we could gather till now. And a Paris test actually increases the actionability of any genomic reports. What I mean by that the next generation sequencing or the DNA sequencing on an average can only match a patient with a drug only about in 18% of cases. Whereas in case of Paris test, it is 4 times more actionable compared to the DNA sequencing. Almost 70% of cases, we could identify a top drug, which can be given to the patient, and it is pan-cancer. And for Paris test, it is more complementing the next generation sequencing. So next, I would like to go through our test process. So we basically we have to get a fresh tumor sample. So the tumor sample can be obtained either via surgical resection or by ascites collection or the pleural effusion, or from the core needle biopsy or punch biopsy. and we require the fresh tissue or the fresh sample come to Syngene within 48 hours of the collection. So once we receive the sample is purified and the organoids were grown from the live sample. And when our organoid team thinks that it has been expanded enough to go through the next process, the drug screening process, we again subject it to the custom-built drug design panel for each cancer. And our robotics technology actually drug it, and a 6-day drug screen assay is performed on the patient screen. And after 6 days, the screen is read, and our proprietary analytics rank the drugs, which again gets curated by two expertise or the two reviewers. One is myself, and another one is our CLIA lab director. So every report gets reviewed by two experts, and we give the ranking for each drug, and ultimately that report gets delivered to patients' oncologist. So by that time, oncologists know by the drug ranking that which drug would be best to choose for the patients. So our drug rankings are based on a score, which is called SPM score. SPM score ranks from 15 to 1. So meaning the 15 to 14 are more of the exceptional responses. So we rank the drugs along with the score, also the category. So basically all the drugs are categorized from exceptional responses to no response. We had exceptional responses, good responses, moderate, low, and no response eventually. So every drug, either it is chemotherapeutics or targeted therapeutics, is always ranked in the same manner. And as we test almost 43 drugs per patient screen, so you can call it that Paris test is analogous to almost more than 40 clinical trial per patient, because we do test every single drug for each patient screen. So we have presented the data in some recent meetings. Last year in the AACR 2020 annual meeting, we have presented our data, as well as the Cholangiocarcinoma Foundation meeting, annual meeting. And in the American Hepatopancreatic Biliary Association of 2020 meeting, there was an oral presentation from our team. And in recent days, in the ASCO meeting, the ASCO 2021 meeting, we have presented a pan-cancer cohort of 419 patients. From 419 patients, we have collected 513 samples, and 628 Paris test screens were performed, meaning a lot of samples were done a repeated screen. For many of the patients, we had repeated screen. So just to give a small snippet from our ASCO poster this year, that we had at least 7% of our total pan-cancer cohort samples as cholangio samples. So we had a total of 42 samples were cholangiocarcinoma samples. And many of our patient samples are of advanced disease stages, meaning clinical stages 3 and 4. And majority of the patient have already seen multiple lines of therapy and progressed. And our median turnaround time is about 28 days, but then for the analysis, we have used all the data from 2016 to till now. Since then, we have revised the turnaround time. If we just take into account last 3 years turnaround time, it is about 3 weeks, which is a very clinically relevant time frame for our drugs to know that if it has activity or not for a particular patient sample. And we have a drug library of almost 220 drugs, which encompasses major cancer targeting pathways. So to give a brief overview of the case of cholangiocarcinoma patients standard of care field is, as you all know, that the approved therapy choices are limited, unfortunately, in the disease space. And the most common standard of care choice for unresectable metastatic disease is gemcitabine and cisplatin combination. For intrahepatic bile duct cancer, we have capecitabine and capecitabine combinations. For if the disease progress, then FOLFOX, FOLFIRI, or regorafenib are the most common choices. We also have some targeted therapies approved, like, for example, FGFR translocated or fusion cases. We have pemigatinib, we have infigratinib, the FGFR inhibitors. For NTRK fusion positive cases, we have entrectinib, larotrectinib. For IDH1 mutated cases, we have ivosidenib. And we also have dabrafenib with tremetinib combination for BRAF mutated patient. So in our cholangiocarcinoma patient cohort, this is from our small cholangiocarcinoma patient cohort, we have seen the maximum number of intrahepatic cholangiocarcinoma cases, almost 67.4%. And majority of our patients are of stage 4 patients, where almost 60.9% of samples were received from stage 4 patient. But what is remarkable that even in that advanced disease stages where multiple therapies have failed before, the Paris test could identify at least one exceptional drug choice in 75% of cases. So we could still see response in certain drugs, which probably were not think for this disease at this phase. So this would be very important to keep in mind for later slides. So as I already mentioned, that we could accept the sample either via surgical resection or a core needle biopsy or ascites sample. So what we have observed that 45% of 45.6% of our samples are from surgical specimen, and we have a very high success rate of organoid derivation from the surgical specimen. It's almost 76%. So important thing to keep in mind that if any patient is undergoing surgery, then that might be best time to send a sample for Paris test, because that's kind of the best opportunity. But we could also, our next best success rate is through ascites sample. We are very successful even growing organoid from ascites sample as well. So the lowest success rate is through core needle biopsy. It also because sometimes we don't receive enough material. We at least need 4 cores for the core needle biopsy. Sometimes there have been cases we only receive 2. So it kind of like get compromised if we have not received the required amount of samples. So we have both treatment naive and pre-treated specimens in our cohort. So we accept both the samples. So the next slide, this is a little complicated slide, I understand. And I want to draw your attention to more of the color. So I hope that you all can see a bright blue color, kind of like already scattered throughout this heat map. So what does that mean? Every blue color or greenish blue color actually denotes an exceptional response. So here I'm showing data from almost 25 patients. Each row is a drug. Each row represents a drug, and each column actually represents a patient sample, a single patient. So as you can see, even in the advanced stages, out of 25 patients we tested, 19 of them had at least one exceptional drug. As if you can see this small red box I'm pointing out, there are two patient data here. Both of them had blue boxes around a certain class of drug, which is RAS-RAF MEK kinase. So both of them had exceptional drug choices in this class of drugs. On the contrary, if you see just next to it where I'm pointing out, it's all yellow. Yellow means no response. So it, as I mentioned before, that it is very, very patient specific. So when we see response to a certain patient sample, there are cases where we don't see a response. So it's not like all across the board, we are seeing the same drugs coming up multiple times. The same way here, another red box I'm pointing to, it's a single patient's data. You can see that all of these are blue or light blue, meaning it has exceptional good responses. And on the contrary, I'm showing it to the right, another yellow box, which means no response. So it is again very patient specific, but you could still see at least one exceptional drug in majority of patients. And our drug library is a very comprehensive list covering all major cancer signaling pathways. So the example I showed just now was for targeted therapy. So what about chemotherapy? So for chemotherapy, as we all know, for cholangiocarcinoma disease field, there are very limited options. And most of the time, the patient progressed, majority of the patients progressed on chemotherapy. And that's what Paris test confirmed, that generally we see a very insensitive nature to most of the chemotherapy, with an exception of gemcitabine. So that's good news, because a lot of patients still receive gemcitabine. And again, every column is a patient, so every box here denotes a different patient. And we could see across the board, there are multiple patients where the gemcitabine is still showing exceptional to good response. So here I'm showing a small expert of our big cohort data, that out of 6 cholangiocarcinoma patients, when he did a retrospective analysis, we could see out of 6 patients, only 1 patient still had some response to the gemcitabine, but all other chemos you see here were insensitive, and Paris test confirmed that we don't see any response to these chemotherapeutics where patient has progressed. So now I'm going to present one of our exceptional cases here. So this patient, we received a sample in 2019, towards late 2019. And the patient has already progressed on multiple therapies, multiple chemo or standard of care, such as capecitabine, gemcitabine, and cisplatin combination. And the patient cancer was FGFR fusion positive. And the patient was able to get into a clinical trial of FGFR inhibitor, which is pemigatinib. But unfortunately, the patient progressed, patient cancer progressed on that. So due to an innate resistance to FGFR-directed therapies. So when we received the sample, our data could confirm that the patient had resistance to FGFR-directed therapies or FGFR inhibitors. But through Paris test, dasatinib came as one of the top exceptional drug. And the patient was able to receive dasatinib along with the bevacizumab combination. And dasatinib actually controlled patient's ascites and pain significantly. And it also extended her survival for several months. Patient was alive at least for 9 months since she started dasatinib. So if we delve deep into her case, into the patient's case, that we saw many of the drugs where she progressed on, such as gemcitabine, capecitabine, or fluorouracil, showed resistance in this sample. And like artefitinib was also FGFR retested. The patient was not sensitive due to innate resistance to FGFR-directed therapies. On the contrary, dasatinib, which was the top drug, it showed a SPM score of 15, which meaning exceptional response. Along with dasatinib, we could also detect multiple other drugs, such as some HDAC inhibitors, velinostat, entinostat, mTOR inhibitor, everolimus, and other drugs, indicating again the same point I told before, that even advanced disease stage where multiple therapies have failed, the patient could still have at least one choice of exceptional drug. So that kind of ends my presentation. Thanks to an amazing, experienced and dedicated team. And special thanks to our Chief Medical Officer, Dr. Astrid Margosian. I think she is probably here. And also the whole team, our CLIA lab director, Dr. Shalini Pereira, and our clinical coordinator, Maddie, our whole organoid team, our drugging team, and our bioinformatics team. So now for the Paris test sample requirement, I have the whole thing listed here. Anyone who is interested can email through this email ID I'm showing here, care@senginmedicine.com, or they can also contact through the 1-800 number listed below. And here we have our clinical coordinator, Maddie Polastro. So if you have any question regarding the sample collection, collection requirement, or how you can contact, how soon you have to contact, I think those will be the questions she would be the best person to answer. And with that, I am ready to take any questions.
Sure. Maddie, thank you for answering some of those questions in the chat, but if you don't mind, I'm going to repeat them just so people who might be listening on Facebook or aren't reading the chat will know the answer as well.
Sure.
So the first question was, please expand on the options for collecting a valid sufficient sample, especially if it is difficult to get a sample via biopsy or if a surgical sample is too old. Would a ctDNA test like Signatera's work? Maddie, do you want to jump in and answer with your response?
Sure, yeah, absolutely. So a kind of nuance to the Paris test is that it requires fresh live tissue. So it needs to be collected 48 hours before making it to our lab here in Seattle. So that means that any archival tissue, unfortunately, would not be a good sample for us to use for Paris testing. Signatera, I believe, looks for circulating DNA in the blood. And while that can show if there's any known mutations in your tumor, we test the tumor specifically to see what drugs your tumor or your tumor organoids respond to the best. And then we rank them and then give that report back. So it's a little different than Signatera. And we also then require tissue rather than blood.
Thank you. And then, is the sample from ascites the liquid that is drained during the paracentesis?
Yes, it is. And if you get a thoracentesis, that is pleural effusion is the liquid that is drained from that. So we accept both of those, and they're my favorite sample to collect simply because they're pretty easy to get and they're pulled for palliative purposes. So I don't have to fight pathology for that sample.
And so fluid that is on the lungs or fluid that is around the abdomen can be used for this testing, correct?
Correct.
Okay. And then which cancer centers participate in this type of testing?
Yeah, so we currently have a clinical study ongoing at the University of Washington. So that's looking specifically at newly diagnosed cholangio patients, and at the time of their surgery, we're taking a piece of that tissue and performing a Paris test on it, and then delivering that back to the oncologist in the event they ever occur. Hopefully they never do, but if they do, then they'll have the Paris test as their kind of plan B. And we meet monthly with that team at a tumor board to discuss the progress of those patients. However, if any oncologist can order the Paris test. So if you are not newly diagnosed or at the University of Washington, I've worked with oncologists all around the world and country. Dr. Javle is one of our champions, among others, of course, but that's just the one that comes to mind.
Okay, and then I just want to clarify, the only place that the trial is at the University of Washington?
Oh, I stand corrected. Sorry. As of this week, it's also being offered at University of Illinois, Chicago. So with Dr. Shika Jain.
Okay. Okay, so for those who can't participate in the clinical trial, how much does this cost and how do they go about doing that? Because insurance does not cover this, correct?
Correct. Yeah. So we currently are not covered by insurance. I've had a few very motivated patients get reimbursed for our services. I have, a one pager. I can help if that's a battle you would like to fight. I'm happy to help fight that with you. But otherwise, the cost of the test out of pocket is $3,500, and that is due once the report is delivered. So if anything, if we're unsuccessful or whatnot, a discounted price is then charged, and all of that is kind of figured out once the report is actually delivered. So
since
no surprises.
Yes. So since Melinda asked about the University of Washington, so for the trial, there is no charge to the patient to enter when it's part of the trial. But if the patient cannot enter the trial for whatever reasons, again, I think the trial is designed for patient naive with resectable disease, because we obtain mostly from surgeries samples, and maybe Maddie correct me if but I believe that that's one of the requirements. And the list price would be $5,000. So we have applying a special discount through the Cholangiocarcinoma Foundation to offer the test at $3,500. And again, if the test is for some reason we're not successful, we will not charge because, as you've seen, the success is not 100%. So something to keep in mind. And it's very, very important that the tissue is adequately collected and it arrives to us in 48 hours. And amazingly enough, that our coordinator worked very hard. They can talk to the physicians, they can talk to the pathologists, and most of the time they make it happen. So do not hesitate. Really, the first thing to do is to get in touch with Maddie and her team.
Wonderful. Okay, so as part of the trial, you're looking for patients who probably are newly diagnosed and operable and having surgery, and then they can have the testing done. And those results would be held for a backup plan in case there's a recurrence or, to move forward that way. But ones who cannot enter into the trial, they can do it on their own, and they would have to do a form of a biopsy or the fluid to send in to you to get the results. And I think a lot of these questions are very similar, so hopefully we answered some of them. And it doesn't matter how much treatment they've had ahead of this, but they do have to go 3 weeks without treatment before they do the biopsy. Is that correct? Somebody want to elaborate on that?
Yeah, so we understand that, especially with our late stage or metastatic patients, that 3 weeks is not always achievable. So if you're progressing on a line of treatment, you are considered eligible for the Paris test. In order to get as much, live tumor content as possible, we try to get a little bit of washout. So that's really up to me and the oncologist on figuring out what is feasible for washout. But our recommendation is 3 weeks.
Okay, perfect. We've been looking into Wiesenthal and Nagorny chemosensitivity testing. How does Sengen differ? Does anybody know that answer?
Understand what was the name of the other test?
Wiesenthal and Nagorny chemosensitivity testing.
Oh, yes, yes. So our test is different because we explore a larger number of targeted therapies. And also we have developed a very unique analytics that enable to accurately predict that involves machine learning and other methods. So I think that is unique to Sengen, both the number and the span of targeted drugs that we can test at once, and also the unique analytics that has a strong predictive power to match the patient with the right drug.
Okay.
Almost on the, sorry,
go ahead.
sorry, Melinda. Almost on
go
the
ahead.
same note, somebody is also asking how it is different with foundation compared to foundation. So maybe Carla, if you would just.
Yes, so yes, foundation is a test based on DNA sequencing. So it provides a blueprint, if you wish, of the tumor. I actually have here maps of DNA sequencing from various cancer types. And the picture of the results of DNA sequences gives potential interpretations on potential therapies. But again, it usually helps a minority of patients. We presented, Dr. Chatterjee presented about 20% of patients can be matched. But again, that match is not proven. It's the correlation. By directly testing drugs on patient live samples, we either confirm the results, and often DNA sequence will say either there is nothing actionable or there are multiple choices. So it helps really to confirm that correlation from the DNA sequencing test to have the Paris test. And in case there is no actionable results from the DNA, the Paris test has the chance of finding actionable therapeutics, even if at this point of time, DNA cannot recognize those genetic lesions. So,
okay.
And that's about 70% of the time we can see actionable therapies.
Okay. I'm just going to emphasize that no insurance companies do not cover this mostly. I mean, Maddie said there's been a couple that have come through, so I encourage you guys to submit it no matter what, just to see. But typically it's not covered under insurance. And then the 43 chemo drugs tested for cholangiocarcinoma, or is that for all cancers? You are testing 43 drugs on cholangiocarcinoma, is that correct?
Yes, selected for cholangiocarcinoma.
Yeah, it's for every cancer, we have different panels. It's cancer specific panels. So for cholangiocarcinoma, it's not only chemo, it's chemo as well as targeted therapy.
How long has the Paris test been in use?
So as we have been, the research has been decades of research. We have been CLIA certified in 2017. So right now we've done hundreds of tests across over 30 tumor types.
Okay.
Yeah.
There is another question which I would like to address. It's about the IDH1. We do have patients which had IDH1 mutation. The only challenge, we could do a drug screen definitely there. The only challenge is the IDH1 inhibitor does not work in vitro system very well. So we do see a weak response, but that might not be kind of an indication that that is not going to work. But we definitely can test all other 43 drugs with the IDH1 mutated patient. We have, I think Maddie can say to this, but at least 2 or 3 patients which had IDH1 mutations.
Is the response ranking on drugs based on cytotoxicity or growth inhibition or the organoids? Well,
so the response, it's based on how the organoids respond to the drug. So it does involve the organoids, and we use an assay that is the most adaptable for the high throughput test that we have. And again, I can give you more scientific reason, but it's a test based on luminescence and the ability of the cells to produce ATP, which has been the best assay to really derive accurate results of drug response.
Okay, and a little bit more information about the clinical trial. Is it just for the testing? Whoops, I lost my question. Hold on. Is it just for the testing, or is there treatment as well? And does a patient have to be physically at either of the two universities, or can they be remote? Well, if they're having surgery, I would imagine. Or can they send the sample to the universities? I guess that's a valid question.
Yeah, that's the question. I feel that they need, I'm pretty sure they need to be at the university. So we would need to see if they would make an exception, but usually the surgeries are carried out at the University of Washington. And there is so it's not just the Paris test that's done for these patients, it's also a liquid biopsies to allow to monitor the patients through treatment that's done in collaboration with Tempus. There is the full genomics also of the initial tumor. So there is a whole series of tests that are carried out.
Okay.
Yeah, so I think the two sites will be University of Washington or University of Illinois in Chicago. Yeah.
Does a severe negative reaction to gemcitabine preclude a patient from pursuing further chemo treatments?
That's a question for an oncologist, so I will not answer that question. Yeah.
They want to know why insurance doesn't cover the Paris test. I think that is the golden question.
Yes, we are working very hard. And actually, I wanted to say when this statement came up that insurance doesn't reimburse yet. But in fact, we have plans, and cholangiocarcinoma is our top priority to advance our conversations with insurances and payers, and we have engaged now expert firms to guide us through this process. So we are hoping to have at least in a year or two being able to offer the test with insurance reimbursement.
And what is the best way for patients to tell their oncologist about this or get the information to their oncologist?
Maybe Maddie, would you answer that?
Yeah, I have a one-pager that has all of our eligibility criteria and whatnot laid out. I can share this with Melinda. Maybe you could circulate it if that would be okay. But the best way is to let them know that you're interested in drug sensitivity testing and to have them contact me at care@syngenmedicine.com. That's our email. They can also give us a call, and really it's just getting that oncologist on board and seeing how we can figure out a procedure and get that sample to our door.
So we have a brochure that explains the Paris test for physicians. So I think that could be informative. We can also provide results summary from our ASCO, direct them to our ASCO presentations, so the physician can see the the depth of the studies that we have carried out to date. So we can provide that information to the doctors. Yeah, but yes, the best way is Maddie and our clinical coordinators will send a lot of material, both directed to the doctors and to the patient via email. Yeah.
Okay. Yes, if you get that information to me, I'd be happy to share it with anybody who wants it. One more question on ascites. Does the ascites sample work if your results have come back as atypical in the past? I guess you talked on how prevalent the ascites works. It still does a good response rate most of the time, right? I remember seeing in the slide.
Yes, yeah, it would.
So, yeah, I think if, you know, let's say that if a pathologist report will say that the ascites might not contain a high percentage of tumor cells, that could potentially affect the results of the test. But we will say, let's try anyway, because we have nothing to lose by trying. So until we receive a sample, we might still be able to derive organoids from it. Yeah.
A patient asking about maybe collaboration with other hospitals, universities be possible in the future for remote support for those who can't travel to those universities?
So yes, so for sure, we are open to collaborations. Again, the main issue is to identify funds to support those collaborations, because again, we have only limited, obviously, we're working across many cancer types. So, but we can offer a discount extended to all these institutions. That's for sure possible. And often institutions have funds for precision medicine. So I think we can talk to at the institutional level and establish collaborations that can then offset the cost to patients.
And one more question about a comparison of the Paris test versus the CLARIS test, CLARIS.
I just did a Google search. Yeah, I'm unfamiliar as well. Yeah. So I'd have to do a little bit more digging to get back. It looks like they do screening if I found the correct one, but let me do some more research.
Yeah,
okay.
We can get back to you, maybe Melinda, with an answer, and whoever asked the question. Yeah.
Yes, I took note of that, and yes, if you can get back to me, that would be great. I'll pass it on. I think we answered most of the questions. If there's any more, feel free to type it in. I think we just got one more. Oh, Caris Live. Maybe they're meaning Caris. Caris is like foundation medicine, right? Foundation one.
It's similar to foundation medicine. They have perhaps more additional informations derived always from biomarkers or expression of certain proteins on the tumor. But again, they are correlative type of informations. They are not direct measuring of drugs. And so the results are generally more limited. And they are always a correlative type, which means there is less less evidence, if you wish, of any treatment. Yeah. So I would say the Caris test is very informative, and the Paris test will add additional information and potentially confirm some of the indicated therapies. Yeah.
Okay. The ascites samples can be sent. They need to be They don't need to be collected. I lost that question. Sent, or do they need to be collected at the institutes? So ascites can be collected whenever they're drained and sent off, correct?
Yeah.
So that's why Maddie likes it, because it's an easier process to have that done. And most patients are doing it, like you said, for comfort and palliative care. And I don't think, I think there's a question in there, but that's more about genetic genes, or I think hereditary questions for patients. And I don't think you guys don't do testing for that, correct?
Correct. Yeah. One thing, if I could just say, to make sure how I think of it at least, is Foundation 1 and Caris, they test the tumor for certain mutations. So in cholangiocarcinoma, FGFR mutations are common. And they might list off all of the FGFR therapies that are approved in cholangiocarcinoma. What makes Syngene unique is the Paris test can then test all of those FGFR inhibitors and see which one is the most, or your tumor is the most sensitive to. So we use that as a guide. If you don't have your genomics, that's okay too, because we know kind of the landscape of what is typically common in cholangiocarcinoma. But in 42 drugs, we're able to also, kind of cover all of our bases. So hopefully that kind of helps with some of those questions.
Yeah, and to me, that's where I see this as really being helpful for the FGFR patients, because there's so many options out there for them, and knowing which one, is going to be more beneficial. And if there's more than one, then that gives them more than one option. So I
Right.
like that.
Rather than kind of going through and trying them, one at a time. So we can help kind of figure out which one works the best sooner.
If you receive a sample and run the test, but new drugs become available later, can those samples be retested against the new drugs?
So if there is enough sample, we save the sample frozen. So I think if it is within, and we biobank for a certain amount of time, but so it could be feasible. It hasn't happened to us. Usually what we do, we obtain a new biopsy also because the patient has undergone new treatments, and we can't assume that, so we need to retest, and it be ideally retesting the sample that's obtained most recently from the patient. Yeah.
There's a couple questions asking about if a patient would benefit from the Paris test. And I guess in my mind, I see it that there's always an opportunity to benefit, knowing if there's a response to one of those 43 drugs as a possible treatment plan. And then would you please say more about palliative care support benefits? I don't know if I confused them about the ascites.
So I think the benefit would be that if the Paris test identifies a drug that's active and promising, and that can be well tolerated by the patient, it will be, it has the chance of improving the ascites and having good benefits. So I'll say to never give up. We just had a press release for an ovarian cancer patient that also had very limited therapeutic choices. We found a targeted therapy that worked very well. The patient 1 year later is doing well. And again, at that point when we received the samples, there was no hope. So
That's great news.
Think that, yeah.
Maddie, will you clarify how soon does the paracentesis fluid have to be sent off after collected?
Oh, yeah, great question. It has to reach our lab within 48 hours. So if you're getting regularly pulled, we can get something on the schedule for next week or later this week, whatever works for you, works for me. It's just, we really need the oncologist's involvement. They have to sign the order form so that the results will go to them. So that honestly is just the probably slowest part. But once I get that, yeah, I'm ready to go.
Okay, and again, to emphasize, you do not use frozen biopsy samples, you want fresh.
Correct.
Okay.
I see this question, if the foundation genetic found no mutation, can you help the patient? Yes, that's a very important application of the Paris test. When genomics says there is nothing actionable, again, that's we can still identify actionable therapies. Yeah.
Wonderful. And you do blood tests for recurrence later on, chemosurgery, now chemo again. So no blood test, just tissue and fluid from either the lungs or the abdomen.
Yeah.
I think, did we get them all? Let's see. Do other comorbidities make a patient an undesirable, sorry, I can't think of another word, candidate for Paris assessment? Comorbidities don't play into the results at all.
Yeah. Yeah.
Okay.
No, I mean, we always will take into consideration if a patient's not indicated for chemotherapies, right, that we might alter the panel so that, you know, we don't waste precious spots with drugs that they can't go on, or if they can't swallow. We really try to keep that in mind as well, but anything else, I don't think, right, Carla?
Okay, good. I think we got to all the questions. A lot of them were answered at the same time. If I missed any of your questions, feel free to email me at the foundation and I will pass them on and get the answers and return them to you. And I think Rick put the care@senginmedicine.com in the chat. And Maddie will send me the one pager. So if you want any information, I could pass that on as well. Just feel free to email me at Foundation. Thank you guys very much. I get a lot of questions about this test from patients all the time. So this is very helpful. And this is recorded, so it will be available on our YouTube channel to share or to rewatch as needed. And again, thank you guys so much for taking the time to present to us today.
Thank you.
Thanks for having us.
Thanks to all the patients. Yes.
Thank you everyone for joining.